Welcome you all once again. So here we are going to discuss the second part of paper Summer Emerges 2020 Biochemistry Paper 1. So in that the question is question 3b, define biotransformation, briefly describe hydrolysis and reduction reactions with two examples. So what is a biotransformation? Biotransformation is another similar word for detoxification. But मोस्ट ऑफ टाइम व्हाट वी राइट हम क्या लिखते हैं वी राइट डेफिनेशन ऑफ detoxification that is wrong. Biotransformation, yes, it is a word similar to detoxification, but sometime what happens, the some less toxic substances may convert into highly toxic substances by biotransformation. It is not like a detoxification. कभी कभी लेस टॉक्सिक कंपाउंड कन्वर्ट होता है हाईली टॉक्सिक कंपाउंड में, उसको हम कहेंगे बायो ट्रांसफॉर्मेशन. देन इट इज नोन एज अ लीथल सिंथेसिस. यहाँ पे लीथल कंपाउंड बनता है, इसलिए हम इसको लीथल सिंथेसिस कहेंगे. The compound produced by this mechanism is more toxic, more mutagenic and carcinogenic than a parent compound. जो parent compound है उसके हिसाब से ये जो compound रहेगा ये रहेगा more toxic, more mutagenic and carcinogenic than a parent compound. So this biotransformation, it is commonly used for the drugs that drugs may convert into the, ideally it should be converted to less toxic, but that compound may convert it to more toxic, and that is responsible for different types of effect or toxicity. Besides, the biotransformation is also called as a biochemical defense mechanism, as it handles different types of xenobiotics, drugs and toxicants और body waste. इन सबको ये handle करता है. So it is the mechanism where the conversion of less toxic compound to more toxic, and that may be responsible for the side effect, and that is it is responsible for mutagenic effect or carcinogenic effect as compared to the parent compound. Okay. So then the next question was describe hydrolysis and reduction reaction with the two example. So यहाँ पे hydrolysis, it is a chemical reaction in which addition of water splits the toxicant into the two fragment of or smaller molecule. इसमें क्या होगा कि hydroxylation होगा, that is incorporation of OH group into the one fragment and hydrogen atom in a which is incorporated into the another part. यहाँ पे H2O में से एक OH जाएगा एक फ्रैगमेंट को और दूसरा OH जाएगा दूसरे फ्रैगमेंट को. ओके, सो इन दिस रिएक्शन द esters, amines, hydrazines, amide, glycosidic bonds and carbamates are generally biotransformed by hydrolysis. So यहाँ पे esters, amines, hydrazines, amides and glycosidic bonds are transformed by hydrolysis. Examples like a drugs जैसे कि aspirin, which is a commonly used painkiller or analgesic, which is a detoxified by the process of hydrolysis. Hydrolysis से ये detoxify होता है. So the enzyme required for the hydrolysis is the esterase enzyme that converts aspirin into the acetic acid and salicylic acid. एस्पिरिन का अगर हाइड्रोलिसिस हम करेंगे तो हाइड्रोलिसिस के बाद ये बनाएगा एसिटिक एसिड एंड सैलिसिलिक एसिड. Similarly, the second drug is atropine. Atropine, which on hydrolysis, it yields tropine and tropic acid. Okay, this is about hydrolysis reaction. Next one is a reduction reaction. So in this reaction, the reductase enzyme is used which contains cytochrome P450 and फ्लेवोप्रोटीन्स. यहाँ पे रिडक्टेज नाम का एंजाइम यूज होगा जो कंटेन करता है साइटोक्रोम P450 and फ्लेवोप्रोटीन्स. And the compound which are detoxified by these reduction reactions, these are the nitro compounds. Nitro compounds इस reaction से यहाँ पे detoxify हो गए and These are reduced to their amines. जो नाइट्रो कंपाउंड्स है वो reduced into their amines form. While aldehyde and ketone bodies are reduced to alcohol. यहाँ पे after reduction of aldehyde and ketones they are reduced into the alcohol. The examples are picric acid, which on reduction it gives picramic acid. Picric acid है जो reduce होके बनाता है picramic acid. Similarly, para-nitrophenol, which get converted to para-aminophenol. These are the two examples of reduction reaction. Next question was differentiate any five characteristic of normal and tumor cells. दोनों के difference यहाँ पे हमें लिखने हैं. So the normal cells and tumor cells. Normal cells are divided into the regulated manner. The division is a well regulated. While in case of tumor, the growth is uncontrolled, uncoordinated, and purposeless. The division of the tumor cells are uncontrolled, uncoordinated, and purposeless. In case of normal cells, show contact inhibition. Here there is a contact inhibition is there in the normal cells, while the tumor cell do not show contact inhibition. यहाँ पे कोई contact inhibition नहीं रहता है. While in case of normal cells, they remain confined. The normal cells remains confined to the particular site, while the tumor cell they do not remain confined in the area of formation. जहाँ बनता है वहाँ पे वो confined नहीं रहता है. They can transfer from their original site by the process of metastasis. Normal cells that undergo differentiation. There is a proper differentiation of normal cells, while tumor cells do not undergo any type of differentiation. ये कोई differentiation में नहीं जाते हैं. This normal cell that can communicate with nearby cells for the homeostasis purpose. They contact their neighbors for the homeostasis purpose, while the tumor cells they do not communicate with the near cell. Nearby cells से communicate नहीं करते हैं. The normal cells they have a characteristic size and shape. They have a proper size and shape, while these tumor cells they have a altered size and shape. The size and shape of cells are not proper as compared to the original cells. The nucleus are comparatively small and light in color. यहाँ पे nucleus whenever we see in a microscope they are small and light in color, while the cells of nucleus cells of tumor cells their nucleus is larger and darker. यहाँ का nucleus थोड़ा large रहेगा and dark रहेगा in microscopy as compared to normal cells. Normal cells, a defined number of well-arranged chromosomes are seen in a normal cells. अगर हम क्रोमोसोम देखेंगे तो यहाँ पे जो क्रोमोसोम है they are well defined, while in case of tumor cells an abnormal number of chromosomes are arranged. There is an abnormal number of chromosomes are arranged. Normal cells they have a defined arrangement and boundary. Normal cells they have a defined arrangement boundaries. While tumor cell the cluster of cells without a boundary. यहाँ पे कोई boundary नहीं रहेगी as compared to normal cell. Tumor cell को boundary नहीं रहेगी, normal cells को boundary रहेगी. The energy जैसे कि normal cell 20% of energy by glycolysis and 70% by Krebs cycle. जो normal cells है तो energy use करते हैं from by the process of glycolysis and 70% it is from Krebs cycle. While ट्यूमर सेल्स ऐसे हैं जो सिर्फ ग्लाइकोलिसिस ही रन करते हैं, उनके पास क्रेब्स साइकिल का कोई रोल नहीं है. नॉर्मल सेल्स एज दे डू नॉट स्प्रेड एज वी नो, while tumor cells they have ability to spread. Okay. Then next question was a case that the 8-year-old female child present to the OPD with angina. On clinical examination, atherosclerotic changes were seen. On investigating her blood report revealed the following conditions like blood parameters value. The random blood sugar was 94 milligram percent, serum cholesterol 432 milligram per deciliter, LDL cholesterol 384, VLDL cholesterol 32 milligram per deciliter, and HDL cholesterol 36 milligram per deciliter, and serum cholesterol TG it is a 104 milligram per deciliter. So what is your probable diagnosis? Explain the three mechanisms for this increased LDL levels and name the drug of choice used for treating these diseases. The answer was for the probable diagnosis is it is a case of it is a defect in a LDL receptor and it is a case of familiar hypercholesterolemia. As we can see that here there is elevated TG, elevated cholesterol level, normal TG that indicates that patient is suffering from familiar hypercholesterolemia. It is a disease of LDL receptor where there is a mutation in a certain LDL receptors result into the abnormally high values of LDL. यहाँ पे जो LDL की value है that is on higher side. Okay. Then what are the three mechanism in which there is an increase in LDL? So here what are the three different mechanism? कौन कौन से अलग अलग mechanism है that is responsible for increase in level of low density lipoprotein? उसमें से पहला है type 2 diabetes mellitus. इस condition में क्या होता है? There is a insufficient insulin production or there is a limit of cell to utilize because there is a insulin resistance is there. तो cell कोई response नहीं देगा insulin को. That's why there is a limitation of your body to ability to process your LDL. Second one is the liver diseases. Liver disease में तुम्हारा LDL बढ़ेगा because liver helps to clear cholesterol. The patient is suffering from liver diseases. The cholesterol clearing will be impaired and your LDL cholesterol will be increased. Similarly, in kidney diseases, kidney play important role in a clearing out of your bloodstream. So the problem if the patient is suffering from chronic kidney diseases that is responsible for increasing level of cholesterol. Similarly, during pregnancy, changes in the body during pregnancy often make cholesterol level high, and that increase is that this condition is increased if the patient is may land up into preeclampsia and eclampsia. Similarly, if the patient is suffering from hypothyroidism, is the low thyroid activity also interferes with the LDL level. So as in case of hypothyroidism, there is a down regulation of LDL receptor also, so that there is a high LDL value often seen in the patient who are suffering from hypothyroidism. तो इस condition में आपका LDL cholesterol level बढ़ता है and the main one is that is a familiar hypercholesterolemia, that is a defect in the LDL receptor that is also responsible for increasing level of LDL. So which drug we can use for the management of this raised LDL? So here we can use atorvastatin, lovastatin, simvastatin, pravastatin, which can be used for the management of this raised cholesterol, LDL cholesterol level. Then next question was describe the cells of immune system. Immune system के cells के बारे में लिखो. So we know that the T lymphocyte, B lymphocyte, natural killer cells, these are the example of immune system. So we are only discussing T lymphocyte, B lymphocyte and natural killer cells. As T cells also get their name from the site of maturation, that is the thymus. T cells also have surface receptor to recognize antigen, but they do not directly bind to the antigen like a surface antibody receptor on the B cell. They can recognize only antigen. It is not just like a B cell. Okay. Now the T lymphocyte differentiate into the two main types. यहाँ पे दो main type में T lymphocyte divided है T helper T cells and cytotoxic T cells. They are generally contain CD4 membrane glycoprotein on the surface. जो T helper cells है, they contain a CD4 membrane glycoprotein on the surface. While cytotoxic they contain CD8 membrane glycoprotein on the surface. ये दोनों का differentiating factor है helper T cell and cytotoxic T cell. Helper T cell that helps or induces immune response. ये induce करते हैं immune response, while cytotoxic they predominantly cytotoxic. Helper T cell recognizes antigen with a class 2 MHC, that is a major histocompatibility complex, and cytotoxic T cell that recognizes class 1 MHC. Macrophages are activated to kill intracellular microorganisms by secreting the cytokinase, while These cytotoxic T cells, they destroy virus infected cell or tumor cells directly. Cytotoxic T cells, they are only for the infected cell or tumor cells, while these helper T cells are responsible for activating macrophages. Macrophages activated to activate to kill intracellular microorganisms by secreting cytokinins. Next one is a B cell lymphocyte. The mature B cell synthesizes and expresses the specific antibodies produced in response to the antigen. It binds the specific antigen by the membrane-bound immunoglobulin or antibody, which is also known as a BCR or B cell receptor. They loses the surface antibody and start producing specific antibody in a large amount to fight against infection. This B lymphocyte produces antibody, hence they have known to trigger the humoral immune response. And the memory B cells are formed after primary infection. They remain in the blood for decades and they circulate in the blood, identify, act against previously infected antigens. And the activated B cell further differentiate into the plasma cells or effector cells. And third one is the natural killer cells. The natural killer cells are a part of innate immunity and they do not have any antigen-specific receptor present on the surface. They play important role in eliminating tumor cells and infected cells. They distinguish the normal cells from the infected or cancerous cell by the MSC class 1 surface molecule, which is absent in most of the abnormal cells. These NK cells are also activated by cytokines known as interferon. This may be an MCQ that natural killer cells are activated by cytokines, that is a interferon. And this activated natural killer cell releases cytotoxic granules which kills infected cells. Then next question was what are the inhibitors and uncouplers of electron transport chain? So जो ETCK inhibitors or uncouplers, this question is for 5 mark. So what are the inhibitors of electron transport chain? That is a complex one between NADH and CoQ. That is the insecticide and fish poison, rotenone, amobarbital, which is a barbiturate, and picaridin A. These are the inhibitors of complex 1. What are the inhibitors of complex 2? That is a trienol trifluoroacetone. That is a ferrous ion chelating agent. Then carboxyl malonate, which is a competitive inhibitor of succinate dehydrogenase. These are the three inhibitors of complex 2. For complex 3, that is cytochrome B, between cytochrome B and C. It is the antimycin A and British anti-levisite. Inhibitor of cytochrome C oxidase, that is complex 4. It is carbon monoxide, cyanide, hydrogen sulfide, and sodium azide. The uncouplers of electron transport chain that uncouples oxidation and phosphorylation, and they disrupt the disruption of proton gradient across the inner mitochondrial membrane, which includes 2,4-dinitrophenol, dinitropisol, FCCP, that is a fluorocarbonyl cyanide phenylhydrazine, and aspirin in a high dose, responsible for it act as an uncoupler of electron transport chain. These are the exogenous one. What are the physiological uncouplers? So in these, the thermogenin, which is a uncoupler, which is responsible for that disturbing that electron transport chain. Then thyroxine, long chain free fatty acid, and unconjugated bilirubin. These are the physiological uncouplers. The separate question may become what are the physiological uncouplers of electron transport chain. Then inhibitors of electron transport chain. The inhibitors of oxidative phosphorylation, it is attractyloside. Attractyloside by inhibiting the transport of ADP into and ATP out of the mitochondria. and oligomycin, which is a antibiotic that completely block oxidation and phosphorylation by blocking the flow of proton through F0 complex of ATP synthesis. तो कौन सा completely block करता है? That is a oligomycin, that is a antibiotic. Define isoenzymes and discuss isoenzymes of three different types. So here you can see that there are physically distinct form of isoenzyme, same enzyme that catalyzes same reaction. They have different molecular form, same enzyme synthesized from same or various tissue. What are the features of isoenzymes? They catalyze the same reaction, but they differ in heat stability. Example: heat stable ALP and heat labile ALP. Okay, they differ in electrophoretic mobility. इनकी electrophoretic mobility different हो सकती है, जैसे कि creatine phosphokinase 1 move faster than creatine phosphokinase 3. They differ in susceptibility to inhibitors. अगर कुछ inhibitor हमने यूज किए तो उसके दो एग्जांपल है टार्टरेट labile acid phosphatase and tartrate stable acid phosphatase. They differ in their subunit. हर किसी के subunit अलग अलग होते हैं, as we have seen in LDL, LDH1 and LDH5. इन सबके सब यूनिट अलग अलग है. ये डिफर इन द टिश्यू लोकेशन. इनका टिश्यू लोकेशन भी डिफरेंट होता है, जैसे कि LDH1 विच इज लोकेटेड इन अ हार्ट एंड LDH5 विच इज लोकेटेड इन अ मसल. दे डिफर इन अ Km वैल्यू. इन दोनों की Km वैल्यू भी अलग अलग होती है, जैसे कि ग्लूकोकाइनेज which is isoenzyme of hexokinase has a high Km value, but hexokinase has a low Km value. तो जब हम isoenzyme की definition लिखते हैं तो in that you have to write they are distinct form of enzyme but catalyzes same reaction. एक ही सब में similarity होती है कि they catalyzes same reaction but they differ into the electrophoretic mobility, they differ in the heat stability, they differ into the susceptibility to an inhibitor, they differ in subunit, they differ in a tissue location, they differ in Km value. ये सब expected होता है. Now here we are going to discuss the isoenzymes of different enzymes. So isoenzymes of lactate dehydrogenase. There are the five isoenzymes of LDH. It is a tetramer made up of two subunit H and M. तो इसमें LDH1, 2, 3, 4 and 5 है. LDH1 which contains four subunit of H present in heart muscle, H3M1 present in RBC, H2M2 present in a brain, HM3 present in liver and skeletal muscles, and M4 present in the liver and skeletal muscles. What are the isoenzymes of creatine kinase? It has a two subunit M and B. Name of the isoenzyme subunit tissue localization CK or CPK. It is B present in brain and it is a BB. CPK2 present in a heart, that is a MB, and CPK3 present in a muscle, that is a skeletal muscle. Then what is the flip pattern of LDH? ये commonly asked question होता है for if there is a case. So normally LDH2 is present in higher concentration than LDH1. Normally LDH2 ज़्यादा होता है as compared to LDH1, but this pattern is reversed in a myocardial infarction. This it has some limited diagnostic importance, इतना ज़्यादा famous नहीं है. तो normally LDH2 LDH1 से ज़्यादा होता है, but this pattern is reversed. In this condition LDH1 is more than LDH2. It is seen in a myocardial infarction. Then isoenzymes of alkaline phosphatase. Alkaline phosphatase के isoenzymes alkaline phosphatase 1, 2, alpha 2 इसमें दो टाइप है, हिट labile and hit stable, pre beta ALP, gamma ALP and leukocyte ALP. So alpha 1 ALP synthesized by the epithelial cells of biliary canaliculi, alpha 2 hit labile produced by the hepatic cells, alpha 2 hit stable produced by placental which is inhibited by phenylalanine, pre beta ALP produced by the osteoblasts, gamma ALP produced by intestinal cells and leukocyte ALP produced by the leukocytes. ये commonly वाइवा में पूछते हैं, commonly asked question for PAQ also. तो ये रट्टा मारना is very important. Then we have a Regan isoenzyme, which is a name after the patient from which it was isolated. This isoenzyme of ALP closely resemble alpha 2 heat stable ALP and otherwise called as a carcinoplacental isoenzyme. What is another name for Regan isoenzyme? It is a carcinoplacental isoenzyme, which is elevated in a carcinoma of lung, liver and intestine. ये जो Regan isoenzyme है, वो cancer of lung, liver और intestine में बढ़ता है. And this enzyme closely resemble to the ALP. Is closely resemble to alpha 2 heat stable ALP. जो alpha 2 है इसके हीट स्टेबल एएलपी को ये क्लोजली रिसेंबल करता है एंड द नेम इज रेगान एंजाइम. ये जिस पेशेंट में पहले मिला उस पेशेंट का नाम यहाँ पे रेगान है एंड इट इज ऑल्सो कॉल्ड एज अ कार्सिनोप्लासेंटल आइसोएंजाइम एंड इट इज एलिवेटेड इन कार्सिनोमा ऑफ लंग, liver and intestine. So this is all about our BAQs. So in next part we will discuss LAQs of summer 2022. Thank you.